Welcome to Under the Microscope. This series is brought to you by the Real Scientists Nano team. Our goal is to provide a platform where scientists can communicate their work and interact with the public. With that in mind, every week we introduce you to a scientist working in the field of materials and nanoscience. Hi everyone, today we have with us Izzy Jayasinghe, who is a UK Research and Innovation Future Leader Fellow and lecturer at the University of Leeds in the UK. Hi Izzy, how are you doing?
Good, how are you?
I'm wonderful and I'm excited to learn more about your research, diving deeper into your research. Let's start with understanding, so how did you end up in your current research field?
I studied biomedical sciences as an undergraduate,
Right.
and at the end of that was in Auckland in New Zealand.
Mm-hmm.
At the end of my studies, I did what they call an honors program. So it's 1 year spent embedded in a research group.
Right.
And the project that I was working on was to understand how signaling, how cells within them carry out signaling.
Mm-hmm.
And I was specifically interested in signals that are made up of calcium.
Right.
And it turned out that the spatial scale in which these signals are produced and they play out is in the nanometer, or sort of tens or hundreds of nanometers.
Mm-hmm.
And one of the focuses of that group that I worked with was to refine microscopy.
Mm-hmm.
To better visualize the signals and the proteins that move the calcium to
Mm-hmm.
enable them.
Mm-hmm.
And that was my introduction into microscopy. And over the last sort of decade and a half, what's happened is that we've moved from one type of microscopy to another.
Mm-hmm.
In pursuit of better resolution, better ability to sort of visualize these proteins.
Mm-hmm.
And that's sort of been my lead into super resolution microscopy,
Okay.
which currently is able to visualize the single proteins that actually produce these signals.
Right.
It's been a really exciting time to be involved in the field.
Right. Yeah, it's just getting better and better, right?
Yeah.
With every passing day.
Yeah.
That's wonderful. That's so cool. Super resolution. Wonderful, wonderful. So, where does your current research fall in this big picture of materials and nanoscience? Where do you come?
So, super resolution microscopy, is done in many different ways.
Mm-hmm.
But I guess the focus that's really close to home for me is single molecule imaging. So imaging fluorescence coming from single biological molecules, proteins.
Mm-hmm.
And so really it's the single molecule imaging that I think I fit into.
Okay, so do you also, while you're imaging, you mentioned you do fluorescence imaging, do you induce some sort of optical indicator? Is that what it's called?
Photoactivation.
Photoactivator?
Yeah. Yes. So it can be either photoactivated or chemically activated.
Okay.
And one of the more recent kind of developments is to actually use DNA as the activator. So you attach the fluorescent molecule to DNA,
Mm-hmm.
and under very specific temperatures, in specific ionic strengths, they can bind and unbind in a way that can mimic photo switching.
Mm-hmm.
So that photoactivation is actually built into the mechanism of how DNA hybridizes. So there's now a variety of different ways of essentially photoactivating or like photolocalizing the markers.
Okay, that's interesting. I hope you're elaborating further on this topic.
I'm gonna talk about this in the on the curator week.
Okay, wonderful. So it sounds to me that you do a lot of interesting experiments. So if you have to pick one from past or present or near past or an experiment or a research project that you're most proud of or most quirky one, could you pick one? I know it's difficult, and explain it to us in simple words in the section we called In Other Words.
So the experiment that I was probably the most excited and proud about is one that I did just before I set up my own group, and that was to apply this new microscopy method called DNA paint, and it stands for DNA point accumulation imaging nanotopography.
Okay.
And so the idea is that you use DNA as markers, and just in my final 4 months of my previous job, I managed to make this work. And the experiment that I was applying this into was about imaging these calcium handling proteins that exist in the heart. And they're called ryanodyne receptors. It's a very giant protein, about 30 by 30 nanometers in size. And it's been heavily studied over the last few decades, and electron microscopy has kind of led the way. But in the process, I became the first person to optically resolve the ryanodyne receptor in situ.
Wow, that is so cool. Oh, how was that moment like when you first time resolved it optically?
I think when you have that kind of moments, you always doubt yourself.
Of course, yeah.
You always wonder if this is an artifact or is this real.
Right.
But I do remember that moment that I realized that this is a different view of the protein that we hadn't seen before.
Right. Wow. So did you have to go out of the lab and breathe a bit?
I remember being really excited that I couldn't sit still. I just needed to tell everyone that this is happening.
Ah, that's so cool. That's what science is for. Like that's one of the perks of doing research.
Yeah,
right?
I hope. I mean, I just wish that it was more frequent. But I think it's a very good feeling to have an experiment that you really wanted and you really invested into. Working is a really really good feeling.
Yeah, it's an amazing feeling. Ah, wonderful. Yeah, definitely going to talk about that as well on
I
the
hope so.
Q&A.
Yes.
Okay, wonderful. So I hope your research experience has been wonderful so far, and it will continue to do so in the future as well. However, if you had 3 wishes to improve the experience, what would you ask for? And I'm not promising anything here.
I think the wish list is that I hope that the way funding is allocated to research will change and will improve. I know that it has improved quite a lot in the last decade or so, but I think if we can maintain that trajectory to make sure that it's a fairer process and that we can retain more people, more talented people, that's the top wish, I would say.
Right.
The second is that I guess in academia we are quite used to kind of ranking the value of the research with various metrics. We have H indices or impact metrics, and that's all very well. But I think we have to. I think we have still quite a long way to go to understanding that is not applicable to every part of science. And that we just need to be kinder to each other when we start, valuing or putting a value on someone's research.
Right.
And I guess the final wish is that, just an opportunity to broaden what scientific outputs mean, that will be a real wish, so that it's not just about writing papers. If people can, it's the scientific community in general can produce preprints and patents and a whole range of things, and can buy into the fact that papers are not the only currency, then that will be a good thing too.
Yeah, all three fair wishes, and they resonate with me as well. I wish I could just do it right away, but I can't. However, I hope we are working towards the better and more improved research experience, so to say.
Thank you.
And speaking of the future, what are you most looking forward to in the next 3 months?
So the thing that's happening in the immediate sort of very short term is that I'm going to start building a new research group in the University of Sheffield.
Okay.
So I'm really looking forward to starting that hopefully in the spring.
Aha, exciting times ahead. Wow. That's wonderful. Congratulations. And I wish a lot of luck and inspiration and good students and amazing equipment.
Yeah, it's beautiful. Certainly a new start.
That's amazing. That's wonderful. And before we let you go, what I want to understand from you is that what are today's nano or material scientists working towards answering? What are the big challenges faced by the field?
I guess there are a number of areas of big challenges. One is, I kind of alluded to this, the lack of equality and diversity and fairness in the way funding is allocated.
Right.
I think funding is sort of the lifeblood of research. So if that process can get better, then that's really important. And so I guess one of the biggest challenges that come with it is, how do you go from being a good laboratory-based scientist to writing good grant proposals to get funding, or approaching industrial funders who will believe you and will fund your research? I think that's a really big challenge for most people, and that has been for me. And that kind of goes in with the issue that retaining skill. If you've trained someone who's really good at one specific thing, and that work is not funded, or if that work environment is not good for that person, then retaining that skill becomes a real challenge.
True.
I think more technically, in my area of research, the kind of the costly nature of the research equipment. I mean, microscopy equipment's quite expensive, and that is a big limiter. So quite often, techniques like super resolution microscopy tend to be limited to really well-funded institutes, and not so much in the hands of people who are not very well-funded. Right. So I think there's a real challenge to try and bring this the cost of this equipment down and make it more accessible. And so there's now a new wave of open source versions of these microscopes, coming online, and so I think if we can keep up with making technology accessible and cheaper, there's a lot of talent out there in the world, not just in the developed world, but in places where funding is the main limitation. Right. If we can overcome that, I think that's a really good thing for science.
Okay. Yeah, that makes sense. I think there is this also, you mentioned this new wave of like tabletop microscopes.
Yeah.
That is coming up as well. Tabletop electron microscopes. This is like the new thing, I feel.
Yes. Yeah. So miniaturizing technology is, I think, the next big frontier. I mean, it's been happening all along. I mean, a really good example is, DNA sequencing of, a protein sequencing,
right,
which has gone from, a bulky machine now to something that is the size of a thumb drive.
right.
So I think the same can happen to imaging technologies.
Mm-hmm. We keep our fingers crossed that we
Yes.
come to that super soon,
Yeah.
in our lifetime at least. That's wonderful. Okay, thank you very much, Izzy, for speaking with us. I enjoyed our talk. I learned a lot, and looking forward to your time on Real Scientist Nano.
Me too. Thank you so much.
Thank you for listening. To know more about us, please visit our website, realscientistsnano.org, and follow us on Twitter at realsci_nano.